inviting me. It's nice to be back. It's been a very long time we haven't interacted, and it would have been really nice to be here in Brittany, but maybe next year. But before, so share my screen. Here we go. First things, so I do understand French, even though I don't speak it very much anymore. So if you have questions or things are unclear, we can discuss in both languages. I wanted to give you understanding that you have a bit of a image processing and engineering background, maybe a view on what this means for something like neuroscience. So how do we use image processing to look into the human brain and to do all sorts of fun things and interesting science with it. So I'll try to introduce a bit all of these concepts, and then I'll go into some of the more technical details. If you have questions on things that are unclear, maybe you can also ask or chat about it early on if things are not working well. But first thing first, so what I study is the human brain, as shown in this beautiful drawing from the anatomist Rudolf Neuhaus, who's actually one of our neighbors in Amsterdam. And here you can appreciate already that it's a quite complex organ with a lot of different pieces, lots of numbers, lots of different things. There's a few things I will talk about, so I'm just going to give you a very, very brief neuroanatomy lesson. So the fluffy stuff on top is the cerebrum. It's often called the cortex, even though it's just the cerebral cortex. You have another cortex here, which is the cerebellum. You have a few other cortices in a lot of different places. And in the middle, the stuff that's inside is called the subcortex. So one thing that I found really fascinating with brain as a research topic is that it goes in a lot of different directions. You can look at brains in terms of evolution. You can start looking across all the different species and how the brain changes in different trees. In that particular study that I'm highlighting here, that was a very simple question, which was asked is how many neurons do different animals have? And so you can see that here they just plotted a lot of different animal species, and then the number of neurons, and the one in H is the human. And so one thing they discovered in that rather complex state of study is that we just have big brains, which is nice, but there's maybe more things. Another thing that I found really interesting is that we can also connect again with evolution, but looking also at genetics. So I hope by now you know that we all have some Neanderthal genes. That's part of us. But those genes are thankfully usually downregulated in terms of gene expression. And in this paper, they looked particularly which genes were, which regions of the human body were downregulating those genes most. And lo and behold, all the ones in blue that you see are brain regions. And in yellow, you have the testes, so for the reproduction. So you can see already that brains are definitely have a lot of interesting structures that are very strongly related to who we are as humans. I also went from evolution, also looking at development, because there also you have a lot of complexity. So the mammalian brain, which you can see here on the left side, it's a fairly simple plan. Everybody has the same, all the mammals. It's built in the same way, and it already has a lot of details in it. But then those things then map in very interesting way in the human brain. For instance, a structure that we call the amygdala that is supposedly related to emotion turns out to be a conglomerate of structures from a lot of different regions of that prototypal brain that then come together in different places. And then one step further, you can then look at all these things together and in adult brains, looking at the timing of myelination in different structures of the cortex. So here the regions that are darker have been myelinating earlier, so getting axons that have been covered in myelin so that connectivity between neurons is optimized. And that also correlates nicely with the myelin density you have in adults in regions here, for instance, very dark. You have a lot of myelin. There are also regions that myelinated first. Those are, for instance, the primary motor and sensory areas that are related to movement and sensation in the body. The one in the back here that was missing on those studies, but it is also very dark, is the visual cortex. And then you can start looking at these things that kind of look a little bit to be the same, which makes sense. It seems to be both myelin growth in some ways. And then you can correlate that with how functional regions of the brain are talking to each other. What is the connectivity between two regions of the brain? And you can see that those maps are actually fairly similar. And then you can just pile it up. You can say, okay, so we have myelination density. We have those functional connectivity gradients that have a lot of things in common. And then we can create maps based on studies that are looking at different aspects. For instance, on the top, one that is looking at the association between brain areas and words that have been heard through podcasts, for several hours of podcasts put inside the scanner, from a level of abstraction to concrete words. And another one which is doing the same thing with movie watching, so watching long movies, long sequences in the scanner, looking at what part of the brain associate with long or short time series. And you can see also pattern there. So that's one thing that I find really crazy in brain science, is that you can just hop back and forth between all these different fields, whether you're looking at evolution, genetics, you're looking at how people process language, anatomy, connectivity, all these things together. So I hope with that, I convinced you that brain science is interesting. One thing that is even more interesting for us engineers is that this also comes from a lot of those results come from a machine that is pretty cool. So the MRI machine, the magnetic resonance imaging magnet, it's a fairly unusual system, because what it does is that it takes the fact that protons inside the hydrogen atoms in your body tend to have that nice spin that would just spin around. And if you put them in a big magnetic field, so here the magnets we're looking at from 1.5 to 3 Tesla to 7 Tesla, and now we can go to 9.4 Tesla. Those are several thousand times stronger than the magnetic field of the Earth, for instance. So when you put them in those fields, they tend to align with the field. They just stay there and they rotate. And then what you can do is you can just push them around back and forth with some radio frequency pulses that have a specific frequency that matches their rotation. And then you can get them out of equilibrium and then they come back. And the cool part, so this is kind of like weird, you're dealing with moving the magnetic field of things inside your atoms. The nice thing is the way those come back then changes depending on the type of tissue you're working with. So looking at different type of relaxation to the equilibrium gives you images of the brain. There's a lot of complicated stuff to actually get the localization as well. I won't talk about it too much, but it's kind of an interesting device to measure things with. And so that's what we get in the end. We can get images like this one, which is a quantitative T1 map of the brain. That's actually an average across, I believe, 30 people who have been co-aligned together. And you can appreciate that there's just a lot of details that you can look at. It's just pretty, right? So one step further, once you have all these images, you can start doing more scientific things. You can start trying to measure things quantitatively. So one thing that has been happening recently in the field, and I've been working quite a lot with, is Quantitative Mri, which is the idea that rather than just get images with weighting of the different type of relaxations, you can get multiple images together and then use a physical model to invert and then get the quantitative parameter. So here you get the relaxation times, proton density, magnetization transfer time, the second relaxation time. And here, if you have diffusion, you can also look at things like anisotropy or diffusivity. One step further that people are very active on right now is how to get from those physical model to biophysical model. So how do we know from those numbers what amount of myelin or iron or what fibers we have in the brain? So there's a lot of modeling going with that. There's a lot of microscopy also to make this happen. But once we have those bridges, we can start using MRI as a way to quantify things that happen, for instance, in development or in disease. Another cool thing with MRI is that it can capture energy consumption because it is sensitive to iron concentration and to the susceptibility of molecules with iron. And hemoglobin versus its, when it's oxygenated or deoxygenated has a different susceptibility. And so if you image someone who is doing a task, for instance, looking at a flickering checkerboard, which is extremely painful to do, but is very efficient to get brain activity in your visual cortex, then you can see things like that signal differences where basically your signal goes up and down with the stimulus being done. And so that's the basis of functional MRI, where we can make tasks and then make people do something with their brain and then look at the blood, the changes in oxygen concentration in the blood via this system. Of course, it's a fairly derived way of looking at function. We are not measuring neuron spiking, for instance, but it's been showing a lot of different things. It's just a very cool way to get data on activity in the entire brain at once. Another fun thing to do is also to look at diffusion. So if you think of the brain as just being mostly water, which is kind of what it is, with a bit of membranes around it. So hydrogen protons, which are very free in water, will tend to move around. And then when they find a membrane, they get stuck there. And with some spin echo MRI, where you're adding some gradients in different directions to knock down some of the signal, then you can start modeling how the diffusion of water can be impaired in some directions, which means that you can then go back from these diffusion model to saying, well, there might be some linear structures. And from those linear structures all the way to creating fiber tracts and kind of anatomy of the white matter and of the connection of the brain, which is also something that is very, very complex and very interesting. So we have all these beautiful images. We get a lot of interesting EMR physics in order to get there. So we need to use things like diffusion, blood oxygenation. We can look at the different type of MRI tricks, and we can get some beautiful images. And so that's pretty much it, right? No, that's really where the work starts, because we really need to get from those images to neuroscientific understanding. And to get there, that's really where, at least I find, that image processing and imaging methods are very, very important, because that takes just a bag of voxels, sometimes usually a pretty big bag of voxels, and then turn them into something you can interpret. So there's a few different kind of flavors of the type of work we're doing in that field. But I try to make some very general categories. The first one is image registration, which is the art of taking images of the brain, different images that have different contrast, different properties, or come from different people, and then put them in the same space to align them as much as possible. You can think that it's fairly easy when it's the same image of the same person from the same session, but sometimes there's distortion of those images, and if you have multiple people, it can be complicated, especially when you have people with disease. Likewise, one thing that's important is to go from voxels to objects. So in order to have objects in your images, you need to label them. You need to segment your images. You need to parcellate some of the structures. You need to find the relationships between them and all that. And you need to model all these things together, which can be quite complicated because you cannot just use images, but you also need to have some information about the anatomy behind it. Another field that we often forget, especially in days where now we have deep learning methods to do everything, but we still have some interesting needs for things like detection and filtering. So how do we, for instance, extract vascular trees from brain images? How do we detect lesions? How do we find tumors? Things like that are very important. And a last field that's a bit more abstract and maybe more focused on neuroscience side of neuroimaging is that we want to be able to map all that rich information that we obtain in those fields into spaces that we can manipulate and work with. So one example is those kind of flat, inflated brain structures where you take all the convolution of the brain and you just smooth it out so you see everything nicely. You can also make other diagrams where you do some dimensionality reduction with various techniques and then plot your data into those different dimensions. So that's also another field that's quite interesting because it links a little bit with visualization, with mathematical modeling, and so on. So let's see. All right, so we have a bit more time, but not a lot. So I have, so I wanted to give you a bit of a flavor more concretely, so what type of work we do. So I have 4 papers from me and my groups at different times where we looked at all those different type of activity. But we'll see, we'll start with a few of those and then see if we want to go further or skip and maybe go to the discussion. So the first one I wanted to bring, it's interesting because it's a very basic image processing problem. You have an image, you want to denoise it. Simple. But we don't want to lose resolution because this image, so in this case, it's a 0.5 millimeter resolution image of the brain, takes a lot of time and a lot of effort to acquire. And so if you start smoothing with doing some Gaussian filtering or something like that, you're basically reducing that resolution that you spent a lot of time and effort acquiring. So you're throwing away the data that you wanted to get in the first place. So how do we do that? How do we reduce the noise without losing resolution? It seems like a simple problem, but there hasn't been really a good solution for it until about 8 years ago now, when this paper comes out from José Mangeon and Pierre Coupé with the idea that you can use in quantitative MRI, especially in diffusion MRI, the fact that you're acquiring data that is similar but not the same across multiple times. So when you do a diffusion MRI imaging experiment, you're putting someone in the scanner, and then you're imaging their brain for diffusion with different angles of diffusion being knocked down. So you can see in those images here, you have the main diffusion, and then you're removing some diffusion in some direction. So here you can see that the diffusion in that direction is very much attenuated. And so you do that for sometimes 60, sometimes 200 different directions around the sphere. And then you get those images and then you combine them to create diffusion models. But before that, so you have a collection of 60 or 100 or 200 images that are the same brain in the same location with very similar contrast, but some differences. And so what you can do is you can take small patches inside those brain images and you just stack them along the contrast direction. And you create a matrix that in one side has the patch the patch voxels, and the other side the contrast. You do a PCA of that. What will that give you? It will give you a series of eigenvectors and eigenvalue. The eigenvectors will first show you the regions where you have the most differences across those regions that are systematic, which happens to be the data that comes from the diffusion encoding. And then at the end of the spectrum with low eigenvalues, you will have changes that just are kind of random across the dimensions, which happen to be noise. So then you just have to find a proper threshold, and then you average the patches that are left, and you get some very beautiful denoising without doing any spatial smoothing, which is the key here. So in my case, I've been interested in using that technique, not on diffusion data where you have about 30 to 60 to 100 images, but on quantitative MRI where we have a much smaller number of images. So here it's the example of a sequence called MP2RegMe. which is fairly complicated, but it's using 5 different images, both magnitude and phase, to create a map, a quantitative map of the first and second relaxation times and also susceptibility. So it's nice because it uses all of these images in different ways, and it's acquired in one way. So all the images are in the same space and you don't have movement in between. So how do we apply those methods on those images? The first thing we need to do a little bit of pre-processing of the phase data, because it includes both the phase differences that are intrinsic to the anatomy and a global phase variation that is intrinsic to the scanner. So knowing that information from physics, we can then try to remove the global phase shifts by doing a total variation representation of the phase. And then we subtract that, and then we can get those images that have a lot more anatomical detail and not so much of the global variation. But that also shows that it's quite important to understand how the physics work in order to realize that this image actually is really this image plus some sort of global variation we don't care about. The second difficult part is now how to find that threshold on your eigenvalues, because now you don't have a very big space and you're basically having a very smooth variation. So in the case where you have a good SNR, you will have some sort of a nice jump that will make things easy for you. But in most cases, you have more of a flat direction. So there's a few different options for that. There's people who've been developing random matrix theory based on some obscure and almost forgotten texts from Russian mathematicians from the 60s. But you can also fit a line to your eigenvalues and base a threshold on that. That's fairly robust. Sorry. So this is a bit how it looks like. So you have on top the singular values, which have different values in your data, and then your eigenvectors. And you can appreciate that the first eigenvector has a lot of anatomy in it, a lot of details, a lot of those convolution of the brain. And then as you go down, you get all the way to almost pure noise fairly quickly. So it's been quite nice for this type of data, because here we have the original data on the left was something that our anatomist would refuse to look at and not even work with because it was just too noisy. And now they are happy starting to label structures, brain structures in those. And of course, we did some testing to check that it actually works. So you can see that you increase your SNR in all those images. All right. So now I'm going to switch to something that's a bit more geometry, which is a bit of a different problem. Here, so we are looking at a piece of cortex that has been measured at very high resolution. And so the human cortex is built of layers. So you have 6 layers of different architectures of neurons, and they just grow in different steps. Actually, they start from the top, the bottom ones, and then they populate all the way to the top. And what happens then is that as the brain grows, it also starts folding based on physics and biomechanics of the tissue, so that we can have a very big brain into a very small skull. But the way it folds then means that you have those layers, which here we can see very nicely at that resolution, kind of bending and stretching in different locations. The problem then is in regular MRI, where we don't see those layers in anatomical scales of 1 millimeter, how do we model those cortical layers? So that's work mostly from one of my PhD students, Mirjam Wennert. So she went back to very old school neuroanatomy textbooks, and she found a very thorough study from a German neuroanatomist named Bock from 1929, who carefully looked at microscopy slices of brains in many different angles, looked at the location and sizes of the different layers across the different configuration of bending and folding, and concluded that, roughly speaking, the bending of the cortical layers has to preserve their local volume. So that's a pretty nice feature. And so we tried to then bring that into a mathematical model. So if you take a piece of cortex here with two different units of size, then you draw lines and make some sort of a cone, shall we say. And then you try to make sure that if you have a layer that is in the middle, then the volume on top and the volume below is the same. So of course, what changes in your system is whether you're in the middle of a flat region where those two little area regions are the same, or if you're in a very folded area where one of them is very small compared to the other, and then the ratio then changes. The way you define them is using curvature of the surface. So first you need to get your MRI, you reconstruct the cortical surface of the brain, you estimate the curvature at every point, both in the white matter and in the edge of the cortex, and then you look at this ratios there. And then I don't know if you guys have looked at something very old-fashioned nowadays, which is called level sets. Maybe, no, Ronan doesn't look very, no, okay. So in the old days.
Yeah, not explicitly level sets, but PD formulation for image processing.
Okay, okay, not bad, not bad. So in the old days, when we were started, level sets methods were very popular to model PDEs where you have a surface components. And so it's a rather natural way to model, for instance, brain surfaces. And the nice thing is that you have an implicit representation, so you don't have to deal with meshes and kind of all the complexity of the connectivity there. And you can use it to basically then add forces that will push your surfaces in different directions. And that's what we've been using in order to take that model of volume preserving layering to then push the cortical layers from different steps forward all the way to the cortical surfaces. One of the nice features there is you can also use it to create profiles that go across the brain by back projection. So once you have that, of course, the first thing we want to do is to know whether or not we are actually doing a good job. So we go back to that really high resolution sample where we see the lamination. And then we start a layer somewhere here, and then we see if everything follows. And so far, we're happy with the model. And then we can look at what other models could do. So if you have two surfaces and you want to find kind of a smooth system between them, A very common way to do that mathematically is to use the solution of the Laplace equation, and you get layers and profiles that are all perfectly orthogonal to each other. You can also just use minimum distance to the surfaces, and you have something fairly simple using, for instance, level set representations. But what those do, it's quite different in neuroanatomy. And so you can see it in those very folded part of the brain here. With the equivolume, basically the part that is lower gets expanded, whereas the Laplace equation goes the other way. So it biases very strongly. And so that makes a difference, because if you look at like a single layer in the MRI, for instance, here the stria of Genari, which runs in the V1, the visual primary area of the brain, you can see that if you don't have the right model, you don't find it in the right location. It kind of moves around a lot. So if you want to do some mapping of different structures across the brain, if you don't have the right model, even though they don't seem to be that different, you start creating very nice features that look like boundaries when they're just artifacts of the curvature. So those things seem to be sometimes a little detailed, but they're very important for the interpretation. Because if you look at a map like this as a neuroscientist, you start thinking about boundaries and areas and separations, whereas the reality is more something like this. All right, so I think it's getting a little late. I have a few more minutes, and I will go extremely quickly onto these two problems. So one thing that comes next that I didn't mention, that I mentioned briefly, was image registration. So the idea is that if you have a lot of different images of the human brain, you would like to average them together. And how do you do that in a way that is as precise and accurate as possible? So this is kind of the state of the art. It's an average of 10 subjects obtained with the software FreeSurfer that just merges the cortical surfaces, and you can appreciate it looks a little brainy, it's nice, but there's also a few like blobby areas that look a little weird. So what we worked on was, can we improve on the state of the art, which is a challenging task to do because people don't like to know that it was not actually very good. And the answer is yes, you can create brain averages that are a lot more detailed. In order to do that, you need to combine a few different things, and that also is a theme that comes back a lot, is that you don't just use one source of information, you combine things like the level set representation of your surfaces, functions of the geometry like curvedness and shape index, and then the T1 values. So we saw that in the maps that I showed you just before, that you can see differences inside the cortex across surfaces if you cut it properly. And so we can use those differences to also align different brains. And then you need to look at things with kind of more complex or less complex structures. So the way we've been doing it is you take a brain, you inflate it like a balloon a little bit until it becomes simpler. You co-register that. And then you inflate it a little less, it becomes a bit more complex, and you co-register that, and you keep going. And then at the end, you get a very nice mapping, and we could show that it's a lot less variable than the other techniques that exist. And that is important for neuroscientists in the end, because then we can do things like this, taking those two different projects we had before, the one about layering and the one about registration. We can start making group averages of the T1 relaxation time, which represent myelin content in the brain at different depths. So from the white matter all the way to the outside of the brain. And you can see that some of the structures that are fairly conserved, they also change. And that's quite nice, because then you can also start looking at finding differences between brain areas based on those patterns. And with the last 2 minutes, I just wanted to talk about things that I'm more interested in now. So we've done a lot of things related to that big fluffy thing on top, the cortex, but it turns out that in the middle, in the subcortex, there's a lot of different structures. And it's really hard because they're tiny and a lot of them are barely visible in MRI. So how do we really see them? We need high resolution imaging, so with the ultra-high-field MRI, and we need a lot of anatomical priors, looking at how things are packed. And also you need multiple sources of information, because some of the structures are not visible in one contrast, but will be very strongly visible in the other, and vice versa. So I will skip over the details, but we recently built a tool that does that. And it's a fairly classical recipe. So you have a parcellation problem. You need location priors. You need to know where structures are. You need some priors about the intensities that you're expecting in those different regions. And then you do a Markov or a Bayesian type of approach where you start building posteriors, looking at the different individual voxels, the shape, the location, having maybe some priors about some of the most stable part of the shape with skeletons, then smoothing, and then growing some final posterior at the end. We validated it, it works. But we also have to be really careful, and that's something that neuroscientists are finding very important, is the method biased? So do we have systematic errors that will be always the same in different subject group or age groups? And it's often the case. For instance, older people will have lower contrast between white matter and gray matter in MRI. So you will have a increase in noise in their data. So you have to quantify all these things as carefully as possible. But the final result of this is to then again go towards neuroscience and try to understand how all these different structures that we finally been able to map, how do they change in terms of shape, in terms of volume, in terms of iron and myelin content over the lifespan. So that was it. Sorry, it was a bit of a very swift overview with a lot of technical terms. But I think the important point there is that if you're interested in looking at neuroimaging problems, one thing to start with is to really get interested in the neuroscience and see what problems are important and what ones are not necessarily so much. And that methods have to be very concrete in their applications. One part I didn't really talk about is that if you really want your work to have an impact, is to really create tools that people can use, people who don't necessarily have an engineering background. So you need to spend a lot of time building toolboxes and maintaining them. But I hope I also conveyed that it's a very rich application domain. It has a lot of technology, the data science, engineering, but there's also biology, philosophy, a lot of things that come together. So with that, I am done talking, and I want to know if you have any questions.
Thanks a lot, Pierre-Louis, for this inspiring talk. We may start the discussion phase and questions. There have been one question posted in the chat. Yassir, could you introduce your question? Yassir, are you still here? Nope.
No, we cannot hear you. So I guess the question was, how do you handle domain shift for different MRI scanner, especially when you don't have labeled data from all scanners? Domain shift.
Can you hear me?
Yeah.
Okay, perfect. So so my question was, you need a lot of data labeling normally if you want to do some sort of labeling in MRI scanners. So my question is, how do you handle the fact that if you have a model for one scanner, how do you handle the domain shift if you want to apply it on different data sets from different scanners?
Yeah. So that's an interesting point, and that's one of the contention point between the deep neural network side of things and the more old-fashioned probabilistic modeling side of things. So if you have a deep neural network based approach or a machine learning based approach in general, the tricky part is your learning base and your application base have to be sufficiently related, and that's not necessarily very easy. So probabilistic models have still been more popular because you create a very simplified model of what your scanner is supposed to give you. So it's usually very detailed about the anatomy, because that shouldn't change with the scanner, but it can have a lot of variations or parameters that you can automatically find from different scanners. So if you have a T1 weighted MRI image, you can segment it with different tools in the same way, independently of the weighting itself. So that's one of the benefits, but it means that you need to create methods that will learn some of their parameters on the incoming data.
Okay, that makes sense. Thanks.
Are there other questions in the audience?
Yes, I have a question.
Go ahead.
When you talk about when you did all the prototype in 3D, did you work only with voxels, or you use voxels and 3D surfaces?
So in general, you can do a lot of work with 3D meshes, so a representation of surfaces with triangular meshes, and a lot of people work with that. I personally don't like it very much because there's a lot of bookkeeping that you need to do. And I've been working with level set for quite a while, which is another way of building surfaces. And so I can just go back and forth between those. So if I need to deform surfaces, I will use a level set representation. So I don't have to worry about triangles becoming too small or too long or splitting. And then when I'm done, I then create a new triangular mesh from that.
Okay, thank you.
Would anyone like to ask another question? Maybe. Okay, I might go for a broader one related to deep learning. I mean, you didn't mention too much the deep learning huge wave, which might like overwhelm everything. So I'd like to know your thoughts on this. I mean, I'm quite sure. I'm not an expert in this field, but I'm quite sure that a huge amount of application and ongoing work using deep learning for brain imaging and different applications. So I'd like to know, okay, how you look at it. I mean, is there room still for more old-fashioned stuff, or will everything will become some kind of deep learning based framework at some point?
Yeah, so how we just going to be feeding data to those algorithms and wait for results? It's a fear for all of us old-fashioned people, I know. So in that field, I mean, deep learning has been moving a lot of applications forward, especially in medical imaging. But I'm at an interesting location where it's actually really hard to use deep learning, because I'm interested in data that is fairly difficult to get. So we have fairly small numbers of subjects. We just published a really big database of ultra-high-field, high-resolution MRI of 100 subjects, whereas people using regular MRI can just take a few thousands of the web in a matter of minutes. So there is definitely a data scarcity there. I'm also working a little bit with microscopy, where it's even worse, because you tend to work with one brain. And so some of the deep learning methods will just be stuck because you're obviously extremely biased, even if you just generate fake examples and so on. The other side also is actually linking to Yassir's question, that in ultra-high field MRI, we have a lot of new contrast. We're working a lot with physicists who are tweaking sequences and say, oh, if you take this one and change this, and then you do this thing, you get this new contrast. Now segment the vessels. And so, of course, we don't have manual annotations on those. We don't have a lot of data to work with, and it can be a contrast that even if we train a neural network on it successfully, by the time we're done training, the physicist will have changed, tweaking the system and find a better way to represent the same thing. So we have to kind of go back again. So in this sense, deep learning is a little less enticing for this type of applications, because the imaging moves very fast. The size of the single image is very big, but we don't have a lot of them because we don't have a lot of subjects. So we can't really do a lot of statistics that are kind of advanced statistics like you need for advanced machine learning. There is, however, some interesting fields where you can use deep learning as a way to estimate the algorithms that would model something. So we've seen it, for instance, in quantitative susceptibility mapping, where you can just generate, you simulate data that would be ideal from the physical perspective, and then you train your algorithms, and then you can explain real data from that. This type of approach that is more a way to speed up computation that would be doable with a very complex kind of simulation or generative model approach, and you can speed things up and it's nicer. But for just kind of image processing itself, it's not as good, and it's also something that has a lot of biases, and that worries neuroscientists a lot for reasons that are completely acceptable. Because if you learn everything you know about the brain from a very small subset of people, and you don't know how you bias yourself when you do that, how your model generalizes on something else, then you can make very, very bad inferences.
Okay, thanks, Pierre-Louis. Questions or ideas to share from other ones? I may have another question, but maybe François, yeah.
It was more a comment, and maybe I would like to know what you think about this. Is okay, you have some available data now all around the world, and everybody can apply new machine learning on it. And basically, from my point of view, you can do big bullshit because you don't know the data well. And the issue is that you may find something which is definitely not relevant. And without any expertise, like you said, in neuroscience or in the data, in the physics, to me, you do, well, nothing really interesting. So the question is more not about the field in itself, but do you have any recommendation to the students to, if they are interested in a new field like neuroimaging or any other one, what they should do? Should they really go into machine learning like everybody else, or try to find a different way? What is your point of view for them, if you have any advice?
That's an interesting question. So, I mean, of course, going into machine learning when everybody is going to machine learning is good, because there's a lot of people who want machine learning. So you get a lot of job options. But if you're really interested in some specific application, if you really want to get into neuroimaging and neuroscience and learning about the brain, a better approach is to look first at the type of work, of the type of research you would be interested. Because if you're interested in doing things in humans with MRI, it's very different from doing things with rats and optogenetics, where it's completely different systems that you work with. So learning about a bit those kind of application domains is good. And at least my personal experience too is that nowadays there's a bit more of a wider and wider gap between the neuroscientists who create very complex experiments that create a lot of very rich data, which they don't necessarily have the background and the tools to understand themselves. And then you need methods that are very much tailored to those data. And I think it's both fruitful and fun to work at that interface where you're close to the experiment, you're close to the neuroscientist, and you're trying to understand how they collect their data, what the data is shaped, and build your methods around it, rather than just grab something from the deep learning shelf, paste the data in, and then wait for some results that comes out. I find that less exciting.
This is also the purpose of this lecture, the computational imaging lecture that we do with Ronan. Thanks.
May I ask one question also?
Yeah, go ahead.
I just wanted to know from a way of thinking the problems, because I know that you define what are the operations you're going to do on the image based on some physical or how do you say, or common sense analysis. But is it common to formulate the problems as optimization problems for these kind of things? Or you already know which are the treatment steps you are going to follow? I don't know if it's a clever question or not, but I was actually thinking about that.
Yeah, thank you. So it's actually a very important question. I think I wasn't really pushing that forward, because to me, the answer is obvious. It's everything has to be an optimization problem one way or another. So you have data, you have a model, you try to fit your model to the data. That's kind of however you're doing it, whether you're registering two images and you look at how those images overlap or don't overlap, which you can do in a lot of different ways, but that's how you drive your registration, because if they're already aligned, then you're done. But if they're far, then you need to figure out where to push in different directions. Or if you
because what I was asking, because let's say, for example, you have in the same optimization formulation, you have both your data and the physical aspect that you try to minimize at the same time.
Yeah, so a lot of the problems we look at tend to turn into we have a model that brings a lot of regularization, and then we have data that kind of pushes that model into weird things. So if you do segmentation, for instance, if you segment the different part of the brain, the ventricles, the part that's in the middle that has cerebrospinal fluid in it, it's a tricky place because it's well located in the middle of the brain, but it changes a lot in shape across different people. So for instance, there you would have to trust your data a bit more, where you have good contrast typically, but then you don't necessarily have good priors to work with. So it's that balance of priors versus data and applying it at different structures or different problems differently that's important.